the C to the I to the central of intelligence. Actually, my name is Charity Iyowosa, you get me? So basically, I'll be talking about biotechnology. Yeah, biotechnology is an interdisciplinary course, you understand, that covers a wide range of study. Courses under it could be gene therapy, recombinant DNA, genetic engineering, and sort of stuff like that. So basically, today I'll be talking about gene therapy. You get me? So basically, gene therapy is the science and technology that deals with the inducing or induction of therapeutical genes into humans or organisms that desires a particular trait due to a defect or a deficiency. Yeah, so basically, gene therapy has been there, but based on evolution and changes, it has become something that is now known and basically studied more often from technologies and basically biotechnologists mainly. So I'm going to be sharing a story, most likely, about gene therapy. Basically, okay, the story is about a guy. There is a small boy, not a guy, a small child. The small child, his name is David, but he was actually referred to as the bubble boy because he was actually diagnosed with immune deficiency disease. You understand? He lacked the enzyme and the name deaminase. So because of that, he lacked it. Gene therapy was to take that place in the diagnosing and treatment of this illness. So it was the place of a gene therapist to do that job. Actually, the guy actually died before gene therapy actually was able to know that this is the cure to his illness. They were at the stage of trial and error method and stuff like that, you get me? So basically, the guy died. The reason why he was called a bubble boy was because based on the immune deficiency he had, it was easier for organisms and viruses to attack him and immediately cause death, you understand, due to microorganism invasion. But because of this bubble, he was able to resist these viruses. It was a system. This bubble was more like a system in which he was able to resist these viruses and prevent the harmful. It was more like covered from these viruses from attacking him, because they know that if these viruses should come in, they will cause catalysis, complete destruction. So that was it, and he died. So sad. Years after, a child called Amihapa was later diagnosed with such a condition, you understand. So she was diagnosed with this condition, but due to the evolution and the advancement in this study that has previously been going on, she was taken and this test was carried out. But this test carried out was ex vivo, which we call in vitro method. Her white blood cells, which is a major agent for fighting microorganisms, viruses, and attack basically the immune system. Gene therapy had evolutionized. They were able to know that they could induce adenine deaminase into her cells to enable her get immunity over these viruses and these attacks. So basically, her white blood cells were removed from her body due to careful method by the biotechnologists, gene therapists. Adenosine deaminase was introduced into the retrovirus, where the retrovirus served as a carrier to transmit the desired trait, which I call ADA, adenosine deaminase, ADA, into the cell. And from there, it was introduced into the cell before it was taken back to the child's body, Ami's body. So basically, Ami had been shown to have lived for more than 5 years after the gene therapy was carried out. So it only tells you something, and that thing is that gene therapy, biotechnology, it is still an advancing field that we still do not know where it's heading towards, but we know it's heading towards a better future in hope. Retrovirus and adenovirus have reduced use. Plasmid DNA is now of recent incorporated into the system and is presently being used. The reason why adenovirus and retrovirus are reduced, the use is reduced to the barest, is because these viruses, when they get into the system, they tend to cause localized and random spread of that gene. For example, now these viruses are not specific. They are not specific and they are not systematic. They go in and they affect everything around that area in which they were sent to. The only difference between a plasmid DNA and the retrovirus is that the retrovirus goes, being a virus, it takes the place of the chromosome and the cell, the gene part, and it replicates the desired trait it wants. Thereby, the organism gets that trait. The only disadvantage of retrovirus is that the environment in which it finds itself, it tends to dominate the environment, causing the desired trait in every single cell, every single gene present in the cell. Basically, the trait will be passed around to everywhere. In a case where these traits are not to be transferred to this DNA, to other genes, the virus has been shown to induce human cold. It causes lots of abnormalities. When we talk about abnormalities, we talk about bronchitis. It induces bronchitis. It induces cough. That's what we call human cough, human cold, and sore throat. You understand? So basically, these are the reasons why retrovirus, adenovirus has been replaced in gene therapy. Basically, gene therapy has been an important aspect of biotechnology because in the case of evolution, in the case of development of pharmaceutical products, and as well changing of the human system where these desired traits are not needed in the body because they are not normal, they are abnormalities in quote. So basically, where they are not needed, gene therapy play a major role. The use of plasmid DNA is now used because when it gets into the cell, it doesn't take the place of the chromosome, or you understand, it just stays in the nucleus and replicates its own that is needed, the gene that is needed to provide defense, immunity, and protection for the organisms. So this is basically the end of this video. I appreciate you guys for staying till the end of this video. I appreciate you so much. If you know you learned something from this video, please share your thoughts in the comments. If you know you have something you want to say and you want to add to it, please share your thoughts in the comments. And if you know this video actually taught you something new as well, don't hesitate to share your thoughts in the comments. So guys, I, Lady C, Charity Iyobusa, I am here to stand as a bridge to fill the gap between those of us that does not like reading our books and our books itself. I'm here to fill that gap. So please guys, follow me and subscribe, because updates like this will be coming frequently. And if you know you want to learn something new to boost for your brains, so that you will not just be carrying hair and you'll be carrying something in, please guys, follow my channel. I'm begging, I'm pleading. I just want you guys to follow me and for us to create this kind of bond, this kind of union, this kind of relationship. Please guys, subscribe to my page and see you. I remain your one and only Charity Iyobusa, Lady C to the I to the Central of Intelligence. Thanks guys.